Hi, I'm Sienna Casciato. I'm an undergraduate student at Rutgers Camden, and I work in Dr. Kwang-Wan Lee's lab, which focuses on circadian rhythms. Today I'll be talking about my research called Characterizing Genetic Mechanisms for Measuring Day Length in Neurospora Crassa. First, I have some background information about the circadian clock and photoperiodism. The circadian clock is a biological process that regulates 24-hour cycles within an organism. A related phenomenon is photoperiodism, which is the response of an organism to a change in its environment, such as a change in day length. There are two hypotheses that describe how the circadian clock and photoperiodism are related. The first one is called the external coincidence hypothesis, which is illustrated in the two images at the top. It states that light is needed to regulate the circadian clock so it can respond to seasonal changes. The second hypothesis is called the internal coincidence hypothesis, which is illustrated in the bottom two images. It states that internal oscillators regulate the circadian clock, while the effect of light on the circadian clock is very small. The goal of this study is to use the model organism Neurospora crassa to determine the genetic mechanism it uses to measure day length. This study is important because understanding the mechanism behind how photoperiod affects life on Earth will allow us to improve or maintain human health that is affected by circadian rhythms and photoperiod. This will be especially important in the future when astronauts begin to explore other planets that have a different photoperiod than Earth. Being able to understand how the circadian rhythm and photoperiod affects human life will allow us to develop technology or treatments to keep astronauts healthy when they're exposed to different photoperiods. Our hypothesis for the study was that the ability to measure day length is a multi-gene trait, so we will be able to use quantitative trait loci or QTL analysis that will allow us to identify genetic elements for photoperiodism. We also thought that genes that are known to be involved in the circadian rhythm play a role in photoperiodism. To conduct this study, we performed a protoparesthesia assay, and protoparesthesia is a female sexual reproductive structure in Neurospora. In the protoparesthesia assay, we incubated 91 wild-type strains of Neurospora into 3 different photoperiods. One was long day, which is 16 hours of light and 8 hours of darkness. One is short day, which is 8 hours of light and 16 hours of darkness. One is equinox, that is 12 hours of light and 12 hours of darkness. They were exposed to these photoperiods for 12 to 14 days at a constant temperature of 25 degrees Celsius. We then removed them from exposure and counted the number of protoparesthesia present on each plate. The data from the protoparesthesia analysis was then used in QTL analysis to determine significant QTLs. A significant QTL is a chromosome that likely has the target gene on it. We found a significant QTL on chromosome 5 and also identified 21 genes on the target region on chromosome 5. We performed the protoparesthesia assay on these 21 target region mutants and also on 10 clock mutants because we wanted to test if the circadian clock was involved in photoperiodism. Here I've also included images of the protoparesthesia assay, and it shows that in different photoperiods, different amounts of protoparesthesia are produced. Here are the results of the QTL analysis. The first diagram shows that chromosome 5 is a major QTL, and the second diagram shows that genotypes can distinguish between different photoperiods. We performed statistical analysis on our data for each of the mutants, so we decided to conduct paired t-tests. For both of these charts, it shows the mutants and its p-value when we compared 2 different photoperiods. So EQ means equinox, LD means long day, and ST means short day. The boxes that say failed normality mean that that mutant failed the normality test, so we separated those into a separate analysis. The p-values that are highlighted in purple mean that they were statistically significant between the photoperiods that they were tested between. And also the data boxes that say NA mean that we don't have data for that mutant yet. In conclusion, we identified a major QTL on chromosome 5 and performed the protoparesthesia assay on 21 target region mutants and 10 clock mutants. We determined that 13 out of 21 genes in the target region were statistically significant between at least 2 photoperiods, and 6 out of the 10 clock genes were statistically significant between at least 2 photoperiods. I wanted to note that this is preliminary data, so future work will include repeating our experiments and continuing data analysis. And here are my references. I wanted to thank NJSGC for awarding me the summer fellowship. And I also wanted to thank the members of Lee Lab, Ningwan Su, Harjit Kiara, and William Myers for their support. Thank you for your attention.